So we are gonna go through what you guys need to do for the state lab that you missed. So we're looking at unraveling the mystery of lactose tolerance. What you're gonna have to do is follow. Okay, all right. What you're gonna have to do is follow along with this video as you fill out the notes, or not the notes. I'm sorry, the lab. When you're done with this video, you should be able to turn it in. If you have any questions, you're going to have to let me know. So, a couple of focus questions. While performing this investigation, we're going to use the following focus questions to guide our work. What is the role of genetic material in determining the structure of proteins and the transmission of traits? How can models show that systems of specialized cells within an organism help them perform the essential functions of life? How can the concepts of statistics and probability be used to explain the variations and distributions of traits that are influenced by genetic and environmental factors in a population? And how have technological advancements made it possible to use genetic information to solve problems in society, such as health issues related to genetics? Purpose of this investigation, you'll investigate the genetics of enzyme production and simulate the mechanisms by which traits are inherited over several generations. So something to keep in mind, the human LCT gene codes for a protein, which is lactase. And we know lactase is the enzyme that breaks down lactose, needed for lactose metabolism. The LCT gene is typically turned off in late childhood when milk dependency declines. It is believed that in ancestral human populations, all humans were actually lactose intolerant. Researchers suggest that a mutation occurred about 7,500 years ago, resulting in a new allele. Individuals who inherited this new allele continue to express the LCT gene into adulthood and were lactose tolerant throughout their lives. The table below is actually very important. So we're going to look at two different populations. An ancestral population and the new population. Phenotype being the physical characteristics of those populations. So the ancestral population had no mutations. Lactase was not produced, and all the adults were lactose intolerant. But for the new LCT phenotype, mutations were present. Lactase was produced, and adults were lactose tolerant, meaning they could drink milk and consume dairy products. So for our first thing that we did, you missed, we did a test where there were two different test tubes. One had lactase powder in it and milk, and the other one just had milk. That's what this procedure is talking about. So reading passage one, lactose, which is a milk sugar, is a large sugar that can be broken down into glucose and galactose by the enzyme lactase. And actually, this part's pretty important. So when lactose is broken down, it turns into glucose and galactose. These small sugars are then able to diffuse from the small intestine into the circulatory system to be transported throughout the body and used as a source of energy by cells. And that part's also pretty important for later in the lab. Individuals who suffer from lactose intolerance, it is because they cannot break the bonds that hold the two chemicals together, molecules together. Naturally occurring bacteria that is present in all of our stomachs or intestines can digest the lactose and use the products to carry out their metabolism and release waste, including carbon dioxide. When those bacteria break down lactose, they make carbon dioxide, and this is what causes the symptoms of lactose intolerance, which is severe abdominal pain, bloating, diarrhea, gas, etc. Okay. So as we read through read steps 3 through 6, we're going to answer question 2 in your student answer packet 1. You can then continue with the procedure. So this is the procedure that you missed. Basically, if I could draw it, this may not be perfect, but so there's one test tube, and there's another test tube. And in the first test tube, there was milk and lactase powder. And in the second test tube, there was just milk. So what it's going to ask us is, all right, which one will have glucose in it? And that is in your student answer packet 1, which is this, student answer packet 1. So we're going to make a claim about whether glucose will be present in both samples at the end of the experiment, provide evidence from passage 1 in the student directions to support your answer. Well, we look what we highlighted. Lactase is needed to break lactose into glucose and galactose. So there should only be glucose in the first test tube and no glucose in the second test tube. So we could say something like, there will be glucose in first test tube and no glucose in second test tube. This is supported. Passage 1, as it states, lactase is needed to break lactose glucose lactose. Okay. When we did the experiment, what we just said turned out to be true. So if it didn't have the lactase powder, there was no glucose, but with the lactase powder, there was glucose. So use evidence from table 1.1 to support or refute your original claim. You know, we were right. So you'd say something like, our claim was correct. Test 2 with lactate powder tested positive. Okay. If you got to pause this or whatever, just go ahead and pause it. So to construct an explanation based on evidence from the experiment in passage 1 and student directions that the presence of glucose indicates lactase activity. It's kind of redundant, but it's asking us the same thing again. Something we could say is glucose indicates lactase activity. As stated, passage 1, lactase lactose, glucose, and Pretty simple. All right, so this next part, we're going to complete the model below to illustrate how the digestive and circulatory system interact when lactase is functioning properly. In your model, we need to include a labeled drawing of a lactose molecule. So lactose is an enzyme. There's no necessary defined shape for it, but we know that lactose attaches to the enzyme lactase. So we're just going to make like a bubble. And we'll label that lactase. Label drawing of the products of the digestion. So I remember from passage 1, it says that lactase is broken down into 2 things, which is lactose, glucose. And arrows to show the movement of the molecules between the 2 systems. So our body uses glucose for cellular respiration. In the paragraph, it describes glucose diffusing into the blood vessel. So there would be glucose in here. And our body does not use galactose, so that would continue on to the rest of the digestive system. Okay. We need to construct an explanation based on our model to describe the importance of the interaction of the digestive system and circulatory system when lactase is functioning properly. So what does lactase do? Lactase makes lactose into glucose, which our body needs for cellular respiration. As if you guys remember, glucose is needed to make ATP. The digestive system breaks down into glucose for the bloodstream cells. And ATP. Sorry, ATP. Okay. Use evidence from passage 2 in the student directions to explain how a change in the structure of the LCT gene or its control regions may change the ability of the cell to produce lactase and affect how cells carry out essential functions in humans that consume milk. So I'm going to go back to passage 2. So you would have done all this in lab, doing the different milk tests. Okay. The next part in the lab, which you would have done, is take this DNA strand and just for reference, you would set up the DNA strand in the way that it told you. You would get these base pairs. And then the piece of paper will be folded over. And you would notice that the triangle or this base pair would fit with this base pair, this base pair would fit with this base pair, but then there would be an issue here and here with a base pairing issue. We have 1 second. I'm going to go to the bathroom really quick. All right. Going back. All right. So I hope that it recorded all of this. Um, what we've done so far here. If not, I'm just gonna quickly scroll through and you can pause if you missed any part of it. All right, so we were on passage two. Nope, hello, not sharing. All right. All right, so we're on passage two. Again, what we were looking at was this part. You make a DNA strand, and when it folds over on itself, the this base fits in that base, and this base fits in this base, but then this base and this base don't match. And that's what this is going to talk about. So, the human LCT gene codes for the production of lactase. It is active in most newborn mammals who depend on milk for nutrition. About 65% of adults have lost their function of this enzyme. One possible mechanism used to regulate the LCT gene is described below. So the LCT gene function depends on the interaction of multiple regions within the DNA of chromosome 2. The DNA within the coding region of the LCT gene contains the code for the production of lactase. The non-coding control regions function as switches and determine if the LCT gene is turned on or off. Many details of how this occurs are not clear, but researchers have determined that when the control regions physically attach to each other, the molecule that transcribes the LCT gene will be activated, and the lactase is produced. If the base sequences of the two DNA segments cannot fully interact, transcription does not occur and lactase is not produced. So if I look at this diagram, here's controller region 1 and controller region 2. Because they're thousands of DNA base pairs apart, they are not touching. So there's no lactase made on this side. But if I have a control region and the LCT gene base pair touching, lactase is then produced because they are touching and not 1,000 DNA base pairs apart. Researchers have identified a partial DNA base sequence within the control region of the LCT gene that appears to be the likely location for mutation that results in lactose tolerance in adulthood. These sequences show a high degree of variation in the DNA samples taken from the cells of 4 different adult participants. So we're going to analyze, we're going to use the information provided in table 2.1 of student answer packet 1 to compare DNA sequences of individual 1 to the DNA sequences of other 3 participants. Individual 1 has the ancestral gene, and we're going to circle or highlight any differences observed. So let's start by answering this. So use evidence from passage 2 and the student directions to explain how a change in the structure of the LCT gene or its control regions may change the ability of the cell to produce lactase. Basically what the passage said, passage 2 states that if the control regions of the LCT gene are not touching, And there is no lactase enzyme produced. Okay. All right, so you can go through and do this. I'll do this really quickly. You're converting DNA to RNA to an amino acid. The amino acid you will need, well, hold on. You're actually going to need the chart. Um, well, yeah. So actually what turns out happening is um, all the amino acids turn out to be the same. And the amino acids would go as follows. TH LYS TRP YS. So actually all the base pairs are the same. So let's do that. All the amino acid sequences turn out to be the same. But the base pairs themselves turn out to be slightly different, actually. So for converting, this becomes ACC, AAG, UGG, G, and UCA. And there's going to be a couple differences. I'll show you in a second. I'm just going to do this real quick. So the first difference is actually here. It's actually going to be AAA compared to AAG, AAG, and AAG. And the last one is UCA, 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 but UCC. And it asks us to highlight the differences. So there's a difference in the RNA sequence there. There's a difference in the RNA sequence here. So they may be all these same amino acids, but the ancestral gene has a difference in this base pair and this base pair. So based on the evidence in tip 2.1, describe the effect of the mutations present in the coding region of the LCT gene on the structure of lactase. So basically what we had to do, I'll draw it again. you had these bases, and let's say that this was A, and this was A. Let's say this was G, and then you had the changed base pair, which was UCA, or you could put the base pair ended up looking different. So what happened is when the DNA strand folded over on itself, the triangle fit into this, and this little loop fit on that, but then this base pair did not fit this base pair. So what we're going to say is due to the structural DNA base pairing, the DNA strand was unable to fold over itself. To search two, inhibited production lactase. So we made that model, and I signed off on everybody's model, so you can put my initials, J and G. What we found was individual one, are all the base pairs complementary? No, they were not. They did not fit together. So they were not transcribed and no lactase was made. But with the mutation, the base pairs were complementary and transcription happened. So lactase was made. Okay. Using table 2.2, table 2.3 to make a claim about the role of the identified mutation in the control region 1. So, the mutation very clearly caused the person to be able to produce lactase so they can consume lactose. So, the mutation caused base pairs to match in the human LCT gene, which allowed production lactase. Therefore, lactose tolerant. Use evidence from table 2.2 to defend the claim that lactose tolerance is a result of a dominant mutation in a specific region of chromosome 2. What we know about dominant and recessive traits is that the dominant trait is shown over the recessive trait. So the mutation on song 2 caused trait because trait is expressed over the lactose in. All right, and now the last thing that we did is we took alleles and mixed them together. So we know that um, an allele has to have two letters. It can be two big letters. It can be a big letter and a small letter, or it can be two small letters. And this was the homozygous dominant. This was the heterozygous allele. And this was the homozygous recessive allele. What we did is we took, everybody started with two alleles and would find a partner, and then you'd mix your cards together and randomly get two new ones. Everybody started with a big and a little, so it was a 50% chance of having either. So 50, 50. We're going to predict how the percentage of each allele will change after 5 generations if no environmental pressures exist. What we said was the little L allele will decrease because the dominant trait will overshadow it. So the recessive little L allele will decrease. The wheel will will over. Sure. No, doesn't sound right. That's fine. And then we went through the trials, and something that you could put, and I'll take this from somebody else, is this person had these allele types as he or she went through his or her trials. I did a whole class data group. That was a data derived from 11 people. I'm sorry, 10 people. And there were two people that ended with the big allele. There were 8 people that ended trial 1 with the heterozygous allele, and there were 0 people that ended with the recessive allele. And then trial 2 went as follows. Trial 3 went like this. Then we got an average of how many times that allele was seen. This would be 2.3, about 2.3 times. This is about 6.3 on average. This is about 1.3 on average. And we calculated the allele probability in the class by filling this out. We found that this was 4.5 because it's the number of big alleles times 2. This was 6.3. That turned out to be 10.8. And we divided by that by the total number of people, which is 10 times 2 is 20, ending with about a 53% chance to have the large allele as the genotype. And then for the little allele, we had 2.5, which is 1.3 times 2. We have 6.3 for the heterozygous, making this 8.8, which then was divided by 20, giving us about a 43% chance of having the little allele. Okay, so use data from simulation to explain whether your prediction was supported or refuted. The prediction that we made was at the very beginning up here. So the recessive allele will decrease because the dominant allele is overshadowing it. 53% chance, 43% chance. Our prediction was correct, but it wasn't super significant. So we could say something like our prediction was correct. There is a 53% chance of having the big L allele, 10% less chance. Then we instituted an environmental pressure because oftentimes an environment will not allow for a certain genotype to be expressed. That certain genotype is the little l. If anybody mixed their cards together and got a little l, they had to start over. So what this person got was this. As the student went through the trial. Okay. This time there were 11 students. From the first trial for the final generation, there were 5, 6, 0. Actually, that all the way down here is going to be 0 because our environment did not allow for this genotype to be expressed. Uh, and on average, this was about 5, and this was about 6. Then when we filled out the proportions or the probability, it came out to be about 10 there, 6, it's a total of 16 divided by 22, giving a 73% chance of having a big L. And then there was 0, 6, 6, 22 students, which then happened to be a 27% chance. All right, so then we got 2 more pages. Okay, so we constructed a bar graph. In the bar graph, I'm just going to pause this real quick. Okay. All right. So in the bar graph, we provided an appropriate title for the graph, made an appropriate scale, and used different bars to represent each allele. So what we titled it was allele probability. And we went up by tens, but I'm actually going to go up by 5s. I wonder if I can. All right, let's see. 5, 10, 15, 20, 25, 30, 40, 45, 50, 55, 60, 75, 80, 90, 90. So let's see. 75, 7, 65, 60, 55, 20. Whatever, you got the idea. Go up by 5s. Yeah. And we had to make a key. And it was either the big L or the little L. And you can technically make this whatever you want. We made big L stripes. And then little L was shaded in completely. All right. So the first percent was 50-50. So 50, 50. Second percent was 53-43. So a little above 50, a little above 40. The last one was 73-27. So a little bit above 70. 5, 10, 15, 20, 23. Okay. So based on passage 3 and student directions, identify the environmental pressure that would account for the allele percentage observed in the second simulation. So passage 3 is here. So current research suggests that the allele resulting in lactose tolerance began to spread unusually fast in some human populations about 7,500 years ago. Some traits provide a selective advantage. Several explanations exist for why it may have been advantageous for some ancient human populations to maintain lactose tolerance through adulthood. Environmental pressures that affect these populations may result in a rapid change in the percentage of people who were lactose tolerant. The table below shows the characteristics of two types of ancestral populations that could have been present 7,500 years ago. So there's a nomadic population and an agricultural population. The big difference between the two, the nomadic, they hunt for wild animals and meat. They follow herds and they forage, while the agricultural population of the ancient peoples bred domestic cattle for meat and milk. That is a big part of the lab because people started to consume dairy and stay in one settlement all year. But the big thing is like this part, like that's the most important part that we're going to get from this reading is the agricultural population started to drink milk. So it would make sense that a mutation for lactose tolerance would be a benefit because if they started to consume dairy, they're going to hopefully be lactose tolerant. Okay, so we go back to the question. In the question, we could say something like the increase in lactose tolerance is because the agricultural population started to consume dairy. So the increase in lactose tolerance is because the agricultural population pretty straightforward. Okay, two questions, and we're done with part 1. part two, I will make the video soon once you finish part one. Um, but it's actually just questions, so it's a little bit easier than what you see here. So, which ancestral population, nomadic or agricultural, is best represented by the simulation where that included the environmental pressure? So, with the environmental pressure, the people that were lactose tolerant was very high. Um, so, people that are lactose tolerant would come from the agricultural population. So the agricultural population represented by the pressure is the amount, or not the amount, sorry, percent of lactose tolerant people is very high. Assumption and make and defend a claim that the change in the percentage of the population with lactose tolerant allele as a result of the interaction of genetic mechanisms and cellular processes. Long story short, all right. So, mutation um, was a genetic advantage so that humans could get glucose, dairy, ATP, or energy for our cells. And so this is part one. If you need to go back to look at any part, pause the video, scroll back, fill out what you need to fill out. I'll have part two done soon. Okay. All right.